A little chillier out there today. They say 85 by Friday. That's good. I'm in favor. I like warm weather. Okay, so today I'm going to talk a little bit about the last part of DNA replication, and it's actually probably the most interesting for a lot of people. And this is replication of eukaryotic DNA. So all the things we've been talking about so far are prokaryotic, although, as I noted, every replication system has a replication fork. Every replication system uses DNA polymerase. Every replication system works 5' to 3'. Those are universal, okay? But there are some things about replication in eukaryotic cells that are different, and they're really fascinating, okay? So that's a little bit about what I'm going to talk about at the beginning of the period today. Eukaryotic cells, I'll remind you, are the cells of higher organisms like plants, animals, human beings. They include things like yeast, which are also eukaryotic cells. And eukaryotic cells, particularly for multicellular organisms, go through what's called a cell cycle. Okay? We don't see a cell cycle in prokaryotic cells, at least not as pronounced as we see in eukaryotic cells. And this cell cycle is orchestrated. It's very orchestrated, meaning that there are controls in the movement of the cell through the various phases. Okay? The phase that we are going to focus on here is what's called the S phase, or the synthesis phase. And this is the time when DNA replication is going on. Okay? But suffice it to say that DNA replication it precedes a phase where there's growth and preparation for cell division being called mitosis. And most of the things that you see of movies of cells and so forth are actually cells that are in the mitosis phase. That pulling apart of the cells is very dramatic, and it's very, very visual, whereas these others aren't quite so easy to see. All right? So, why do we have an orchestrated cell cycle in eukaryotic cells? Well, in eukaryotic organisms. Well, for multicellular organisms, it makes very good sense. We have to coordinate everything. Okay? Eukaryotic organisms want to have coordination of growth, for example. I want my left leg to be at least approximately the same length as my right leg. I want the symmetry of my body to be reasonable. Nobody has perfect symmetry, but I want it to be reasonable. And that happens as a result of controlled cell division. All right? So these are important things for eukaryotic organisms. If we don't control cell division, we end up with organisms that don't have as nicely functioning parts, as it were. And totally out of control cell division, of course, means that we have cancer that kills the organism. So we want to have control over that. So cell cycle makes a lot of sense for cells to have. Okay? Now, one of the things that happens in eukaryotic cells, of course, DNA replication occurs. And it occurs at sites within the chromosomes known as origins. And so we call them origins like we call them in E. coli cells. Many of the same protein functions that we saw in prokaryotic cells are also present in eukaryotic cells. They have different names, but the functions are very similar. We see things like a helicase. We see things like a DNA gyrase. We see things like the beta clamp. Okay? So all these things, and we see things like single-stranded binding protein. We see these things in eukaryotic cells just like we saw in prokaryotic cells. In eukaryotic cells, they have different names. But there's one thing in eukaryotic cells that we can't get around. And it is this fundamental difference that gives rise to several things inside of eukaryotic cells. And this thing is a linear chromosome. Bacterial cells all have circular DNAs. Eukaryotic cells all have linear chromosomes, meaning they have ends. Well, if you think about replication of a circular DNA, let's say you take that leading strand, you go all the way around that circle, it's eventually going to come back to where it started, right? So there's no end to worry about. And when it comes all the way back around to where it started and it hits that RNA primer, what's going to happen? What enzyme is going to get invoked to remove the RNA primer? No. To remove the primer is catalyzed by DNA polymerase 1. That's the thing that's got the 5' to 3' exonuclease. Okay? So that RNA primer disappears and we have a perfect DNA. What if we have a linear chromosome and we start with an RNA primer at the very end? What's going to come around and remove it? Nothing. Nothing is going to come around and remove it. That's a very, very important point. Okay? The only way that it gets removed is when it comes all the way around, then there's that gap filled in because now we have a pre-existing primer that's come all the way around that can get extended, and we can finish off the polymerization and make a perfect circle. We can't do that with a linear chromosome. We can start it just dandy, but we can't finish it. If we come back and we remove the RNA, and we have things that will remove the RNA, how do we start without a primer? We can't start without a primer. So what that means, and this is a very, very important point, what that means for a eukaryotic cell is that every time a eukaryotic cell replicates, its chromosome gets just a little tiny bit shorter. Every single time. Well, you can imagine that you don't have chromosomes of infinite length. And so if you start taking a little bit away each time, you can imagine that eventually you're going to get to a point where you're going to start doing some damage. You're going to start losing DNA that's going to be important after a period of time. And that's exactly what happens. But what's that period of time? How long does it take for this to happen? Okay? Well, it depends, as many things do. In order to understand this process, we need to understand something about what's at the linear end of eukaryotic chromosomes. Okay? So we're going to say a little bit about those here. Okay? This depicts that process, and if you don't understand what's going on here, I won't bore you with it, but suffice it to say that at the end of each replication, we're losing a little bit at each end. Well, cells aren't stupid. Cells basically know, cells don't know anything, but cells know that they need to have as much of their chromosome as possible. So at the linear ends of chromosomes, eukaryotic cells have something called a telomere, Telomere, a telomere. What is a telomere? A telomere is a sequence of DNA that's repeated thousands of times. It's a short segment, about 8 or 9 nucleotides, and you'll have thousands of copies of those 8 or 9 nucleotides that are end to end to end to end to end to end, all on the end of a eukaryotic chromosome. That's what a telomere is. A repeated short sequence repeated thousands of times. Well, now you start seeing why eukaryotic cells have a telomere. They have a telomere so that when replication proceeds and you lose a few bases, you're losing some of the telomere. By the way, the telomere doesn't do anything. It doesn't code for protein. It doesn't code for anything. It's simply there, and it's there largely so it can be lost without any damage to the cell. Well, okay, that's good. Well, yes and no, until you run out of telomere. So once you've deleted all the telomeres, then what starts happening? Well, now you start chewing into the meat of the chromosome, the place where the genes are that your body needs or your cell needs. When it gets to that point, the cell is hosed. It is hosed. Okay? Now, some people started scratching their heads with this and said, the length of a telomere must be pretty important. The longer a telomere is, the longer time it will take before you start chewing away the goodie of the Oreo cookie. All right? That is the genes that you need within that chromosome. And so they started doing investigation, and sure enough, what they found was something very interesting. They could predict the lifetime of cells by examining the length of the telomeres that they had. Okay? Well, let's think about this. I've got telomeres that are shorter than anybody in here, because my cells have been dividing longer than your cells have, and my telomeres are getting shorter and shorter and shorter. Okay? That's not a very pleasant thought, is it? It's not to me anyway. I'm older than you guys are, right? You guys say, oh, I got a long ways before I got something to worry about my chromosomes having a problem. Well, how did those telomeres get there in the first place? That's one of the first questions we want to ask. How did they get there, and why don't they just get fixed? Okay? Well, the way they get there is by action of an enzyme called telomerase, T-E-L-O-M-E-R-A-S-E. Telomerase is an enzyme that, I hope you're all sitting down, makes telomeres. Of course, nobody fainted with that one. Okay? Telomerase makes telomeres. I'm going to show you in a second how it does that. Okay? And they're trying to show you this here, but it's not a very good diagram, unfortunately. Okay? I'm going to show you how that works. All right? Telomerase makes telomeres. Well, if it makes telomeres, why do our telomeres get shorter? They get shorter because it's only active when we're an embryo. When we're an embryo, that telomerase, when we're a fertilized egg, that telomerase is sitting there going just crazy. It is lengthening those telomeres as long as it can. Well, how does a telomerase do that if a DNA polymerase itself can't do it? Well, telomerase is a very interesting and very unusual DNA polymerase. It still also works in the 5' to 3' direction, but it has a little trick that allows it to extend the end of a chromosome. Okay, and I'll show you that trick in a second. All right? All right. So, here is the end of a linear chromosome. Okay? If I start, as I said before, with an RNA and I go in this way, this guy still can't be replaced or fixed very easily. All right? Now, how then do I make a linear chromosome be longer? Well, it's entirely possible for the other strand to be lengthened. There's no primer, there's no template to copy, there's no strand to copy, but, and this is where telomerase is very special, it carries its own strand to be copied. So it's actually carrying a strand. This green strand that you see right here in this figure is one that the telomerase is carrying with it as luggage on itself. This little segment right here is about 8 or 9 nucleotides long, depending on the organism. And yes, that's the one that's getting copied over and over and over and over and over. That's why we see this thing being repeated. Because the telomerase lays down the template, it makes the copy of it on the other strand, and then it slides down and it does it again. And it slides down and does it again. And it slides down and does it again. It'll do that thousands of times. Well, you say, well, the top strand now is very long and sticking out here. What about the other strand? Well, the other strand, I can now take an RNA polymerase, a primase, for example, make a primer, and then I can start filling in. So the primase will still come in, it will still copy this other strand to get it started. Then DNA polymerase fills in, and we've lengthened the chromosome. We can't get all the way to the very last nucleotide at the very end because we can't fill in that last RNA, but look at all this new DNA that we've added as a result of action of the telomerase. Now, that's a little complicated concept. I understand that. So I'm going to slow down and ask for questions about that. But look what it's done. It's elongated. We see it's been elongated here. Now we see the primase coming in saying, oh, here's something I can copy. I can now fill in. So now this new guy, this new chromosome, is longer than it was up here. Yes, sir? It varies. It varies, not necessarily. Oh, good question. So his question is, are telomeres mostly GC for holding those ends together? Okay. Well, GC you would predict would actually be useful for that purpose, but that's not a requirement for a telomere. No. Okay? But it's a good question because ends do tend to kind of fly apart a little bit. Yes? Oh, very good question. So what happens, so the next thing is, well, maybe I've got the fountain of youth. The fountain of youth would be, if I can make my cells make telomerase, then, bingo, I live forever, right? Well, maybe. There's just one problem with that, and the problem is that there are other cells that have telomerase active, and they live forever too. They're known as cancer cells. Yeah. Somebody had to spoil the party, right? Okay. Now, it may be that we can control this in some way, so I'm not saying that there's no answer, because there are people very interested in doing this, but it tells us that we have to be very careful if we're going to mess with telomerase, because we don't want to encourage uncontrolled growth. In fact, what people are doing right now is exactly the opposite. The opposite being that, well, if cancer cells need telomerase, why would cancer cells need telomerase? They divide uncontrollably. If they don't have telomerase, what's going to happen to them? They're going to have the same problem that regular cells are. They're going to shorten their telomeres, they're going to lose their ends, and they're going to die. So one of the strategies for treating certain types of cancer is to take and use telomerase inhibitors, drugs that stop telomerase from functioning. And in some cases, in some cancers, it appears that they work very effectively. The cancer literally burns itself out. Now that would be good, except for there's one other group of cells that we have to think about. There's all these exceptions. And the other group of cells we have to think about are stem cells. Stem cells are the cells that can differentiate and become anything else. We have stem cells throughout our body. If we just routinely gave telomerase inhibitors, we might be doing some damage to our stem cells. So though this is a very exciting and very interesting area of research, it's something that tells us we're going to have to be pretty careful as we design strategies or therapies that affect telomerase in some way. Okay. Now let me come back to this. Do you have questions about how this is working? Do you understand what's going on? You don't understand what's going on? No questions. Okay. Yeah. Well, I haven't talked about a mechanism for turning it off, no. No, I'm not talking about that. I'm talking about, do you understand how it's doing what it's doing? It's carrying its own primer, it's copying its own primer, and then it just slides down and keeps sliding down in that process. That makes the top strand, the bottom strand gets made then by primase coming back in and filling in the gaps. Make sense? Okay. There's one other thing I haven't told you. And the other thing is, this is a really interesting enzyme. Okay? The nucleic acid, this short 8 or 9 base pair sequence that it carries, is not DNA. It's carrying a little segment of RNA. It's carrying a segment of RNA. That means that what it's copying is not DNA. It's actually copying RNA, and it's making DNA from it. That is, it's using RNA as a template to make DNA. Enzymes that have that characteristic, that is, that copy RNA and make DNA, are special kinds of DNA polymerases that we call reverse transcriptases. Now, this reverse transcriptase copies its own template. Other reverse transcriptases don't. Now, somebody asked me the other day, do we have reverse transcriptases in our cell? Not effectively very much, no. All right? Well, where do reverse transcriptases come up? Well, they come up with RNA viruses like HIV that have to go through a DNA intermediate, and they have an RNA genome. So they copy RNA and make DNA. That's one of the things that reverse transcriptases will do. Okay. So, telomerase is a very special DNA polymerase known as a reverse transcriptase. Very important thing. So it's copying an RNA to make a DNA. That's a good question. Her question is, does telomerase hop from one piece of DNA to another, or does it just stay and keep moving down? It's my understanding, though I'm not an expert in this, it's my understanding that it just stays on and keeps sliding further on down. Yep. Yep. And then eventually what happens is, as the embryo matures, the cells stop making telomerase. So we have the gene to code for it in our genome, but it stops being made. Yes, sir? Not coding for any proteins at all. That's right. So it's basically what some people used to refer to as junk DNA. That name is starting to disappear. People used to use that term to refer to anything that didn't code for protein. And so if you look at eukaryotic chromosomes, you see a lot of what people call junk DNA, because there's all kinds of stuff in there that doesn't code for protein. But we're learning more and more about that it's not really junk. Okay, good stuff, cool stuff. All right, now, I want to say one last thing, and I usually talk about it right here. And I'm not going to talk about the polymerase, I'm not going to talk about this. But it does relate to eukaryotic DNA replication, and it relates to cancer. Okay? So I think this is very important to consider. Eukaryotic replication, DNA replication, is very complicated because, remember, we've got histones, proteins around which the DNA is wrapped. So there's a very much bigger level of orchestration that's necessary to replicate eukaryotic DNA than there is prokaryotic DNA, which doesn't have any proteins on it at all. Okay? The consequences for mutation in eukaryotic cells are very large. We want to avoid cancer, right? If the more mutation we have, the more likely we are to have a cancer arise. So eukaryotic cells have a quality control system built into them that prokaryotic cells do not. Okay? Now we talked about repair, and we talked about those things. Repair is one quality control system, but the quality control system I'm getting ready to tell you about is different than what prokaryotes have. This is in eukaryotes, okay? This is a really interesting system. What eukaryotes have is a system. They have a protein called P53. P53. Just the letter P followed by 5-3. P53 is one of the most interesting proteins in your entire body. Okay? It's the quality control protein. What happens is P53 literally monitors the DNA replication process. And what it's doing is checking to see that the replication process didn't have any problems during the replication cycle. What would that be? Well, let's imagine you're a smoker. And you're smoking, and you're getting some of those big, honking molecules I said get stuck onto a DNA during replication. Or maybe you've been eating some food that has some really nasty chemicals in it that the food industry has been dumping into there, and it's damaged your DNA. Okay? In either case, when replication was proceeding and it hits one of those stretches, I said one of the things that the polymerase can do is it can guess and just move on, but sometimes the block is such that the polymerase can't do anything. It doesn't finish its job. That's what P53 is looking for. Places where the replication did not complete. So when P53 senses that this has happened, it will stop the cell from going on to the next phase. We saw the 4 phases, the G2 phase. It will stop the cell from going on to that next phase. And in the process, what it will do is it will stimulate the cell to start making DNA repair proteins. Those repair proteins come up. So there's some floating around, but now P53 says, whoa, we got a real emergency. Make all the DNA repair proteins that you can, because if we don't fix this, we're going to have trouble. So the cell starts synthesizing all the different DNA repair proteins, and the DNA repair proteins go to the site where the damage occurred. Now, one of two things can happen. One thing that can happen is that the repair proteins can fix the damage, and everything is fine. And when that happens, P53 signals to the cell, okay, go ahead and go to the next phase of the cell cycle and get ready to divide. On the other hand, if the repair proteins, the damage is so bad that the repair proteins cannot fix it, p53 will sense that also, and it will stimulate cellular suicide. The cell actually commits suicide if the damage cannot be repaired. Now, just before class, somebody asked me about, well, why do some people get cancer and some people don't? Is it genetic? Okay. If you smoke a lot and you hear these people live to be 90 years old, is it genetic? Well, in some cases, it's genetic. Okay. In some cases, people have a predisposition to have cancer. Not so much a predisposition to be protected against cancer, but a predisposition to have cancer. One of the ways they can have a predisposition to have cancer is if they have damage to their p53 system. If you lose your quality control, you got problems. Many cancers are known that when people analyze them, they discover that p53 got knocked out in the process. p53 got knocked out. Instead of the cell being able to commit suicide because it couldn't go forward, instead the cell went forward and cancer arose as a result. Okay? So p53 is a very, very important protein in helping the cell to avoid situations of cancer. It's avoiding those mutational type events that would happen from replicating or moving ahead when replication really shouldn't proceed. Okay. Yes, question?
Did you say that
p53 signals the cellular suicide when replication cannot be fixed. Yeah. Pretty amazing protein. Yes? How do p53s get damaged? Really good question. One of the ways would be if you mutated the gene that coded for p53. So here's a prime example where mutation can lead you to a cancer. Okay? So this is why we want to be avoiding what are called mutagens, because mutagens are more likely going to give rise to mutation. And this is the random part. The mutations are random where they're occurring. You sure don't want one of them occurring in your p53. We'll see some others where we definitely don't want them occurring or we've got real problems. So you're rolling the dice. The more mutagens, the more tanning booths, the more times you're rolling the dice. I don't want snake eyes, man. No snake eyes. All right. Other questions? Okay. Well, let's turn our attention now to synthesizing RNA. We've been talking about synthesizing DNA. So let's talk about synthesizing RNA. First of all, you saw the name at the top of the last slide, and that name was transcription. I want you to know what that name is, and I want you to know what that name means. Transcription, underline this, means the synthesis of RNA using DNA as a template. The synthesis of RNA using DNA as a template. What's a template? A template is something we copy. Okay. Now, here's some general things on the screen that I'm going to just lead you through briefly. The synthesis of RNA is similar in some ways to DNA and very different in other ways. Let's start with similarities first. First, synthesis of RNA moves only in the 5' to 3' direction, just like DNA. Only in the 5' to 3' direction. RNA uses nucleotides, but instead of using deoxyribonucleotides, it uses ribonucleotides, ATP, GTP, CTP, and UTP. Now, here's a very important and interesting difference. RNA synthesis does not require a primer. RNA polymerase works just dandy without a primer. It does not need a primer to start things. As I said, it goes 5' to 3'. We'll see that it copies one strand of the DNA, not the other. And we'll also see that the DNA does not get changed in the process. Okay. Well, let's talk about synthesis. Now, first I'm going to start talking about prokaryotic cells again. But a lot of the things that happen in prokaryotic cells, again, are similar to what happened in eukaryotic cells. And I'll point out differences later. Okay? So for right now, we're just going to talk about prokaryotic cells. Well, in DNA replication occurred at a special sequence that we called an origin. That's where DNA replication starts. It starts at an origin. RNA replication starts near a special sequence we call a promoter. A promoter. I didn't say it starts at, but I said near a sequence we call a promoter. Let's take a look at that. What you see on the screen are a whole bunch of different genes in prokaryotic cells. You can see in yellow over here the place where transcription starts. That is the very first nucleotide that the RNA polymerase puts into the RNA. Okay? And you see something in pink and you see something in blue. And what these are related sequences that are all related to each other. Okay? So I'm going to explain these to you. All right. The first of these, this pink one, is called the Pribno box. The Pribno box. The Pribno box has a sequence that if we took all these, look at this, the T lines up pretty much everywhere. The A, there's a G right there, but the A pretty much lines up. There's a T there. All right, but we see similarity throughout the sequence. So if we average them all out, the most common sequence we get is shown down here, the Pribno box. 79% of the time, the first one is a T. 95% of the time, the second one's an A. 44% of the time, it's a T, blah, blah, blah. Okay? This sequence is located in what's called the -10 region because it's about 10 nucleotides away from the place where transcription starts. Remember I said that transcription starts near the promoter. This is the promoter region of the DNA. This whole region here is the promoter region. That's right. All right. Now, look at that sequence. What does that sequence tell you? What did you learn about TA base pairs? They're easier to pull apart, right? Well, one of the things that RNA polymerase has to do during the process of transcription is it has to get in between the strands and copy one of them. That means the strands have to be pulled apart, and voila, here is something that's easier to pull apart than this sequence or other sequences around it. That's kind of cool. The chemistry of the DNA molecule, the cell is respecting and using to its advantage. Now, you say, well, how come they're not all identical? It's a good question. The answer to that is that not all genes are transcribed in the same quantities. They're not all transcribed, meaning that some genes, the cell might make 1,000 copies. In other cells, in other genes, the cell might make 4 or 5 copies. I talked about DNA polymerase 3 last time, and I said there's only 4 or 5 individual enzymes in the cell. Okay? The more RNA the cell makes, the more protein it's going to make. So if you have something that's you don't want to make a lot of, there's no reason to make 1,000 copies of the RNA and waste the cell's energy. You would make 1 copy and that would be it. Well, these sequences help determine how many copies are made. We'll see another factor coming up later. But these sequences help determine how many copies are made. That is, how many RNA molecules are being made. All right? Okay, well, let's think about this. Here is the most common sequence that appears in the Pribno box. What we know is that genes that have this exact sequence, like this guy right here, are made in large quantities in the cell. And those that vary from this sequence, like let's say this guy up here, TAC-TGT, the ones that vary from that sequence aren't made as abundantly. Why is that? Well, there's a very good reason. In E. coli, the RNA polymerase is literally reading the DNA, looking for a sequence. And a sequence like TAT-AAT, it likes. It reads it very readily, it stops there, and it starts transcription. Things that vary from there, it may just go sliding over it and not notice it. Only with low frequency will it stop and start making an RNA. So the sequence really determines how readily the RNA polymerase stops and starts making RNA. Does that make sense? Everybody's stunned in silence here. Okay, I'll say that again. So, the closer that any of these Pribno box sequences are to a TAT-AAT, the more likely the RNA polymerase will see it and stop and start making RNA. That means the more close they are to this sequence, the more RNAs will be made. The farther they are away from that sequence, the less likely the RNA polymerase will see it and stop and make RNA, and therefore the fewer RNA molecules will be made. Question? Okay, very good question. Is this sequence present in front of all genes in prokaryotic cells? In E. coli, this sequence can be found in front of almost, not every one, but almost every gene. We'll see some exceptions, but this is a very, very, very common sequence. Yeah, question? What is it about polymerase that allows for such variability? Good question also. Polymerase is a protein, and proteins recognize, there are many proteins that bind to nucleic acid, in this case binding to DNA, and actually interact with them. DNA polymerase, we saw, for example, interacted and it polymerized and made things in. It had the ability to read the other strand and put in the appropriate nucleotide for it. And RNA polymerase is reading both strands. It's reading the sequence that's there, just like a DNA polymerase is, but now instead of just reading the sequence, and it's actually getting to a point, stopping and opening it up. So it depends on the structure of the protein. The RNA polymerase has a specific structure that its given structure will bind more specifically to certain sequences than it will to others. And that's based on the amino acid sequence that's in the protein. Okay? Okay. Now, what's this thing back here? Well, this is a sequence that's also somewhat similar in a number of cells. It's called the -35 region, and it's about 35 bases ahead of where the transcription start site is. Okay? It's not quite as, and you see there's a little bit of variation in terms of where it actually appears in there, but it has reasonable conservation of sequence. And this sequence also helps the RNA polymerase to recognize, hey, this is a gene, and I want you to transcribe it. That's what both these sequences together are doing. This one's involved in being pulled apart. This one is a little bit more involved in being recognized, but they both are recognized, actually. Okay. A lot of stuff there. Let's talk about the chain of events that happen during transcription. Okay? This introduces another protein that plays a role in the process. Now, I said RNA polymerase slides along and finds a sequence that says, oh, wow, this is a promoter, right? Well, RNA polymerase has help. And it has help in the form of a protein factor called sigma. That little brown guy underneath the RNA polymerase right there is sigma. What sigma does is it helps the polymerase to say, there's the sequence we're going to transcribe. That's what sigma does. When sigma and the protein and the RNA polymerase together find that promoter, they stop, like I said. The polymerase peels apart the strands, as you can see here, and it starts copying one of them. In this case, it's copying the bottom strand. We see what's called a transcription bubble that looked kind of like the replication bubble we had for DNA. Except for now, instead of both strands being copied, only the bottom strand is being copied. And you can also see that the RNA is hanging off. It doesn't stay attached to the DNA. It's actually hanging off like here. So this bubble is moving along to the right. It's moving along. It's making things 5' to 3'. That means it's copying the bottom strand, which is oriented 3' to 5'. Okay? And we see it hanging off up here. Once everything has gotten started, sigma takes a hike. It leaves. So sigma's role is in helping the RNA polymerase to recognize what is a promoter sequence. So sigma's pretty important. Sigma's pretty important. We'll talk more about sigma in just a little bit. Questions about this? Okay. Well, there are 3 phases to transcription. You just saw initiation. Elongation is pretty simple. It just keeps moving along, getting longer and longer and longer. The last phase is called termination. And termination has a couple cool elements to it. Okay? Termination in E. coli cells, E. coli being a prokaryote, okay? Termination in E. coli cells occurs by 2 different mechanisms. One is called factor independent and one is called factor dependent. I'm going to describe both of them to you here. Okay? All right. Factor independent. All right. Here is a DNA being copied. Let's imagine we're copying this bottom strand, and the RNA polymerase has been rolling along, doing its thing, making that strand. Let's say the RNA polymerase is actually right here. Okay, it's just finished synthesizing this guy right here. It's not shown in the DNA, but it's just finished synthesizing this guy. It's copying a DNA strand that's up, that's over here, right? Copying the DNA strand. Here's the RNA that's hanging off of it. Look at this RNA. This RNA polymerase has finished copying its gene, and it's hit a sequence that has something in it called an inverted repeat. Look at this inverted repeat. AAA, GGC, TCC. Now look at this sequence over here, going the other way. AAA, GGC, TCC. It's inverted on the other strand. This gene that this RNA polymerase was copying had something at the end of it that looked like this. Well, the interesting thing about this is the RNA, when it gets copied, all of a sudden goes, oh, wow, look, I can make base pair because A can pair with the A here, compare with the T here, which becomes a U, of course. A with U, A with U, A with U, A with U, A with U. And look at this. It makes something we call a stem loop. This is forming in the RNA as the RNA polymerase is moving along. Well, remember now, this is at the end of the gene. The gene has already been made, and now what this serves as is a signal that this is the place to stop. This is the place to stop. Okay? Well, what happens is this thing forms all by itself. Because these base pairs are so close to each other, they automatically go, oh, I can make a duplex. And they make a duplex, and that's how this structure forms. Now let's think about what's happening. Okay? Here is the RNA polymerase. Before, this was a nice linear piece of DNA, and all of a sudden, this duplex formed. The DNA it's copying is down here. The RNA polymerase is here. The RNA polymerase was lying very nice and quiet, minding itself, and now all of a sudden this thing has jacked up, and it has lifted the butt end of the RNA polymerase up. It's physically lifted the RNA polymerase up. Just like a jack would lift your car, this has lifted the end of the RNA polymerase up, and the RNA polymerase was copying the DNA. What this thing just did was it just lifted it off of the DNA. It physically stopped the replication process, the RNA transcription process from happening. Pretty cool. This method is called factor independent because it doesn't take any other protein to do it. It's simply an RNA that literally stops its own synthesis. This only happens for genes which have this sequence at the end. Not all genes have this sequence at the end. That leads to one of the protein, and then I'll finish for the day. The other protein is a factor. Okay? It's a factor called Rho. That's the symbol for Rho right there. And this one's kind of cool too. Right? Here's this transcription process occurring as we saw before. There's the RNA hanging off of the DNA duplex. Rho has a structure that's a circle that looks kind of like the beta clamp. But it has a very, very cool feature. The example I like to give for the feature is, how many of you took gym in high school and one of the things you had to do was you had to climb the rope? How many people hated that? You liked it? I hated it. How many people liked that? Okay, well, all right. So I hated it, all right? Well, my description is, here's that rope hanging off there, and there is the row, that's you, that's climbing it. And what's happening is Rho is sliding up this thing, and it's trying to catch up with the RNA polymerase. It's a race. So instead of you being in the gym climbing that rope, somebody's letting the rope out as you're trying to climb it, because this strand keeps getting longer and longer. Okay? So if you're climbing that rope in high school and trying to get to the top and somebody is letting it out, you're having to scoop pretty fast to get up there. This guy is having to scoop pretty fast to get up there, and Rho will eventually catch up with the RNA polymerase, and when it does exactly the same thing that happened before. It lifts the RNA polymerase off of the DNA, and everything falls apart. This is a protein factor that makes that process happen. Rho is pretty cool. Rho requires ATP to do this, requires energy, but it's a race. Well, how does it know where to stop? It doesn't. It depends on how fast Rho moves and how slow the polymerase moves. Well, what determines how slow the polymerase moves? I'll give you a hint. It's related to GC base pairs. Polymerases go slower through GC base pairs, and so Rho will tend to catch up with it in the place where there are GC base pairs. So Rho doesn't give termination that's exact at a specific point, but it gives usually a slight range of places where it stops. But it does stop it. And that's cool. Okay, you guys up for a song? Let's do a song. This is an easy one to sing. It's called Transcription. And it goes. Phosphodiesters are the bonds of RNA that support a ribopolymer made of G, C, U, and A. The RNA polymerase binds to a Tata box, I can't hear you, and copies from the template strand all along the way it walks. Initiation of transcription thus proceeds. From the closed to open complexing in the DNA it reads. The sigma factor gets released, its work is over fast. Polymerase can then advance after this step has been passed. In elongation, the polymerizing spree moves along the way in fits and starts synthesizing 523. The RNA is floppy and it dangles from one end. Oh, that's a most important thing for you to comprehend. Then termination finishes the RNAs, thanks to protein Rho or hairpin forms that release the polymerase. So this describes transcription steps in 3-part harmonies. Here's hoping with this melody you can learn it all with ease. All right.